Thank you. Thank you. Thank you for the invitation. It's a pleasure to be here. And I would like to present some approach that we are using in our lab to study dementia using EEG. Well, aging and brain health and neurodegeneration are modulated by different factors related with diversity and disparity. And the approach to analyze the influence of this factor is using a multicenter study, which allow to develop biomarkers sensitive to diversity and disparity, and that can be applicable across different countries. The idea of carrying out EEG multicentric study for us is developing a global model of brain health that can be sensitive to diversity and disparity, using an open science approach. So we try to share data, to share codes, and the results. And just a reminder why EEG, because it's direct representation of the brain electrical activity, and it's a tool that it's easy to replicate, cost-effective, and scalable. We carry out this multicentric study using a large public available data set, or participating in different purpose-driven scientific consortia, like, for example, the EuroLab EEG consortium. This consortium at the moment is not exactly the number of recording that is involved, but there is a contribution of different labs of different countries, mainly from Latin America and Europe, and mainly with resting state EEG data from healthy participants and participants with mild cognitive impairment and different types of dementia. And this is good because we have a lot of data, but also this is a challenge because all the data are recorded using different procedures, including different number of electrodes. So we have to try to harmonize this data to reduce the variability across site. As you mentioned before, there are many harmonization very good harmonization pipelines that are available, and we have developed our own procedures. Here there is like a picture of the different steps that we use to in our analysis. This is the pipeline that is constantly. We are continuously increasing the number of metrics that we obtain, and including also different pre-processing step and normalization step. And I would like to stress one of the topics that you were mentioned before, that this is the dealing with recording with different number of electrodes. In the pipeline, we have preprocessing, a preprocessing block. That this is important because we already know that processing have a great impact on the results that we are obtaining when we are processing EEG. As an example, this is in this figure we show how changing the reference electrode can impact a spectral component of the analysis and connectivity at the skull level, and also connectivity in the source EEG level, and also the estimations of the EEG cortical generators. And one of the steps that we include in this pipeline is transforming one electrode layout to another. and we test different possibilities when we were doing that, and try to reduce the number of electrodes, try to increase the number of electrodes, and try to compute the error when you transform one electrode layout to another one. And we test that using connectivity analysis and at the EEG source level, and we observe even when the data variability decrease, there is always an impact of the result that we can obtain. In this case, there is a reduction in the or an increase in the bilateral connection that we were observing after harmonization, and a decrease in the short length connections that we were obtaining. We have published a couple of papers using this harmonization procedure, and I would like to share with you some of the considerations that we have in the group. This is one of the topics we talk a lot. And at first, I would like to say that first, harmonization is a need. Data harmonization is necessary when for multicentric study, because most of the studies are based on existing data, and they are acquired with different electrode configurations. So sometimes decisions need to be made using a pragmatic perspective. We need to implement harmonization procedure to enable multicentric studies with regional or global impact. And at the same time, we need to consider that data transformation can influence the study outcomes. And one example is the transformation of the electrode density layout when conducting EEG source space analysis. We have to deal with that. We know that reducing the number of electrodes can have an impact on the resolution on source estimation. Otherwise, creating virtual electrode can imply that sources are estimated with a lot of redundant information. So there is a trade-off in the type of decision that we have to make. Also, we need to consider that the impact of the harmonization can vary across EEG metric. So using a particular dose involving electrode layout configuration transformations can have a unique effect on different source space EEG metrics. And for example, connectivity metrics tend to be more sensitive to stuff modification than spectral metrics or complexity metric estimated at the given brain region. Finally, and it is important that even when we believe that we are using the most powerful harmonization pipeline, we need to focus on addressing confoundings. And this is critical for the results interpretation. And about that, our group have systematically expanding the set of control analysis that we use to quantify the residual influence of relevant variables after harmonization. For example, we test the relationship between the electrode number and different sociodemographic variables. The use of signal quality metric as a predictor of regression in different analysis that we are conducting. Also, measuring the association between this quality signal metrics with sociodemographics and all the number of electrode, and also with trying to establish association between the number of participants and sociodemographic variables. This is probably I was too fast. This is what I want to mention. Before finishing, I want to this is the core of the BrainLat group, the close collaboration, and this is the institutions that contribute to our work. And that's it. Thank you very much.